Chameleons are incredible animals that we know can change the color of their skin dramatically. Almost all of you watching this video have at least seen one chameleon, whether it's in person or through TV or any kind of media. And maybe as you've watched this chameleon, you've wondered, well, how did these animals change their color? As I just mentioned, chameleons are known for their dramatic changes in color. It's generally thought that this color change is similar to cephalopods like squid, that have these organs called chromatophores with these pigment-containing structures within them, which allow for a dazzling display of color. Majority, actually all of this video is coming from a paper in the journal Nature, which I'm going to post in the description. This paper presents really interesting insights into how these animals actually change the color of their skin. The main piece is that these animals don't use color changing cells. They use these guanine crystals. And today we're going to talk about that. Now, these crystals are within these structures called iridophores. These are stacked chromatophores, those stacked pigment-containing organs. And there's two layers of these crystals, and we're going to talk about that for the majority of this video. One thing I did want to mention is that throughout the course of this paper, they did discuss that melanin and an unknown dark blue pigment containing chromatophore contribute to some of the colors in chameleons, but not nearly as many as these crystals. Males, females, and young chameleons can display these melanin and dark blue chromatophores, as well as some of these really unique colors. But we'll talk about how the expression of this color really depends on the gender and development. Another side piece, these colors change due to things like emotion, stress, competition, all sorts of other things. This paper used all sorts of really interesting techniques to study these colors, but they also used different situations they put these chameleons in order to allow them to express the colors, to study them further. So we mentioned these guanine crystals, these layers of crystals in the skin. We mentioned two layers. So first we're going to talk about the first layer called the S iridophores, or superficial iridophores. Real quick, these guanine crystals. Guanine is a chemical that's present in all sorts of different things. In this case, this makes up these crystals. These crystals kind of look like this. Most importantly, they are highly reflective, which corresponds partially to the change in color. Some more details here. They have a lattice structure. Think of a lattice like an apple pie design, this like crosshatch type of thing. They are very organized. Now, this is very important because this is one of the big differences between S iridophores and D iridophores, which we'll talk about a little later. And naturally, they are photonic-like. This word has a really complex definition, but from what I understand, we can consider something like an opal that has a natural photonic crystal where light reflects and refracts in all sorts of different directions, corresponding to dramatic changes in color, especially as you move the object. And we think of an opal. You look at an opal, light shines in all sorts of different places and you get, greens, reds, purples, blues. So these guanine crystals have a similar type of property. As I do this whole video, keep in mind that the exact mechanism is currently unknown, but there has been enough research, as you'll see, to really determine how important these crystals are. So these iridophores are fully developed in males. This is directly responsible for their color change. They are present in females and young, but they are not fully developed. So this paper studied panther chameleons, a pretty bright kind of chameleon. They found that S iridophores are directly responsible for color changes and brightness in a significant way. They also determined that shifts in the distance and organization of these crystals really corresponded to the dramatic show that you see when these colors change. And this is a preview. We're going to talk about this in the next slide. But as you can see, first of all, these things are very organized, right? There's layers to them. There's not much disorder going on. And as you can see here, the relaxed chameleon, we see some greens, a little red, some blues. The excited chameleon As you can see, this green turns to yellow, these red blues turn to red, and there's all sorts of different color changes. But you see that these structures go from very close together to very spread apart. This is one of the really interesting findings made in this paper. So let's talk about some specifics, how this change in distance and how light really gives us a dramatic change in color. So the main piece of this paper is really trying to figure out, how did these colors change in such a dramatic fashion? They determined that these crystal structures are responsible for a color change. But what are some of these specifics? So I've mentioned before in other videos, change in color means a change in the wavelength of light. I've mentioned before how light can come in a wave and a particle, but as far as the waveform, a wavelength is the difference between one top of a wave to one top of a wave. When we look at the visible light spectrum, we see that all these colors are perceivable because of their different wavelengths. So for example, this purple wavelength is a lot closer together than the orange wavelength. This is how these colors present themselves. It's all the distance between the tops of their waves. A point they made was that for color to change from blue to red, from this wavelength over here to this, pigment containing structures are great, but they can't really correspond to such a broad change in wavelength. And they found that these crystals are a big part of that, specifically the shift in the nanocrystal distance. You just call them nanocrystals, these tiny crystals. As I mentioned over here from the last slide, the difference between the relaxed and excited state, you can see these organized structures are tightly packed together and then they spread apart. And this offers a different perception for us and the environment regarding the change in their color. So in this case, this would correspond from blue to turn to red, and it causes a shift from green to yellow orange. So from this area to about this area. Let's zoom in here and look and see what's going on. So we have these greens, we have a blue, some yellow, orange, some red. And so what we're seeing here, going a little closer, this blue here turns into a red. Now it's not deep red, but as far as the spectrum is concerned, it's red, orange. This green turns into red, orange. This streak here gets a little lighter, but overall, if I just erase all this, you can see that the colors change very, very dramatically. And again, we have this relaxed male chameleon and excited chameleon. Just take a look and see the dramatic change in color. This green going to a yellow, these blues going to a red. And here we have a different look into these iridophores. So the S iridophores we have up top, this top layer, like I said, and these D iridophores down here. And then we see here these different interesting structures. This has more of a box structure, and this is a hexagonal structure. Overall, the most important piece is observing this dramatic change in the color due to the arrangement of these crystals. So if we go over here, the paper discussed all sorts of different avenues the color paths could go down. I made a little flow chart. May not be the most accurate, but this is what I perceived given what was in the paper. So males can go from a blue to a green. Not a crazy shift in wavelength. We just referenced this over here. But then from green, it can go to white, red, or can go to yellow, orange. And this is when the wavelength can jump up a decent amount. And this is all because of the orientation and the nature of these crystals, how they spread out, how they change the way they look and their form. And this allows light to strike these crystals, showing off some pretty cool colors. And to add, the expansion and contraction of these crystals really allow for a broad show of the visible spectrum of light when it comes to the colors that we can see. As I mentioned before, the exact mechanism is unknown, but they at least identified the culprits for this interesting display. So let's talk about D iridophores. D stands for deep. These are the structures that lie underneath the superficial iridophores. An interesting part about this paper is the team was trying to figure out, okay, what did D iridophores have to do with color? A lot of the paper was discussing the S portion. They found that D iridophores are actually not responsible for visible color changes. This is really interesting. They found that these are large, brick-shaped, and unorganized structures. If we move over here, you can see them. They're large and they have like a brick shape to them. And they're not organized whatsoever. So if we just compare E here to D, you can just pause and see they're nothing alike. The D iridophores are kind of dispersed randomly, and there's no real structure and organization to them. Outside of their structure and the way they look, these things are present no matter the age, sex, or gender. I mentioned before that these S iridophores are mainly developed in males. These D iridophores are present in all of them. While they're not reflective in the visible light spectrum, they're highly reflective in the near infrared region of light. About 45% of sunlight is reflected back when the sunlight moves through the S iridophores and hits the D iridophores. At this point in the paper, a lot of their discussion of these iridophores was a hypothesis. They say that these things potentially play a role in thermal protection. A lot more study is needed. The panther and veiled chameleons live in these hot and dry climates, and at least 45% of that sunlight is reflected. So they're thinking, well, what if, as an evolutionary tool, as a structure for survival, these animals not only had these S iridophores they could camouflage and show their colors, they had these D iridophores as a mechanism to protect from the hot, intense heat. So is this a structure? Are these structures geared towards survival in temperate environments? Since they're not responsible for color, could they be responsible for protection? And outside of the methods of data collection, this is pretty much how they ended off the paper. It's a very, very well written paper. I enjoyed it. So let's go through the summary and discuss what we learned about color and chameleons. We started off talking about how chameleons are thought to change color like squid do, with these organs that contain pigments. And this paper in Nature says that these guanine crystal structures are really responsible for the dramatic changes in the colors of these animals. The first layers of crystals are known as the S iridophores, these guanine crystals that are very organized, very reflective, fully developed in males, are responsible for these major changes in color and the brightness, with the shifts in distance and organization being a huge factor. Then we highlighted that shift in structure and organization, how it allows for dramatic changes in the wavelength and in the visible light spectrum, along with the expansion and contraction of these crystals, gives us a really broad experience when it comes to perceiving the different colors that these animals express. And here it was shown that pigment is only a small portion of the mechanism by which these animals can show off their colors. We then talked about how D iridophores are not responsible for visible color changes, but they could have a role in thermal protection, or protecting the skin in the body of these chameleons, especially those who live in very hot and dry climates. We talked about how about 45% of sunlight is reflected off of these structures. And this led to the main hypothesis being some sort of structure geared towards survival, especially in these intense environments. So that's all I got. I hope you guys learned a lot. This was a very, very fun video to research and write up. I did not know any of this, so it's always fun for me to learn as I teach you guys, so all of us can learn. And as always, thank you so much for watching and listening. Please like, comment, and subscribe. Tell your friends, and I'll be back soon with another video.